Hello friends, today's topic of discussion is translation or protein biosynthesis. Protein biosynthesis. It is one of the frequently asked question in university examination. So it is important LAQ for university examination, and this is a must read topic from genetics. Translation in eukaryote or protein biosynthesis. We will discuss this topic under the heading of what do we mean by translation process, that is definition of translation, then requirement for the translation process. Requirements are all types of RNA, that is mRNA, tRNA, then ribosomes, then amino acids, and factors. Various factors are required, that is for initiation, elongation and termination. Then steps, which includes activation of tRNA, then initiation, elongation and termination. Okay, so what do you mean by translation? It is process in which the codons or genetic information in the form of codons, it is, suppose this is mRNA, this is 5 prime to 3 prime, and these are the codons. So genetic information in the form of codons, these are the triplet nucleotide sequence or triplet base pair sequence present on mRNA, read from 5 prime to 3 prime direction. So these codons are decoded into amino acids for the synthesis of peptide or protein. So it is a process by which the information in the form of codons present on mRNA, it is used for the synthesis, or it is translated into amino acids for the synthesis of peptide or protein. This is translation. Okay, so what is requirement for the translation? mRNA is required which contain genetic information or codons. These are the triplet base pair sequence present on mRNA molecule, read from 5 prime to 3 prime direction, non-overlapping, comma-less, unambiguous. There is no other way to interpret it. Specific, they are universal, and they follow the degeneracy. Okay, degeneracy means one amino acid is coded by more than one codon, that is known as degeneracy of genetic code. Okay, so mRNA is required. Then tRNA, it is adapter molecule. It is an adapter molecule. It is required to carry the amino acid to the ribosomes for the synthesis of peptide or proteins. So tRNA is required here. Then ribosomes in eukaryote, it is 80S ribosome. So this 80S unit, it is composed of two subunits, that is 60S and 40S. So it is composed of 60S ribosomal unit and 40S ribosomal unit. So this 60S ribosomal unit, it is composed of RNA and protein. So it is composed of 28S rRNA, then 5.8S rRNA, 5S rRNA, and near about 50 proteins. While this 40S subunit, it is composed of 18S rRNA and near about 34 proteins. Okay, so this is composition of eukaryotic ribosome, 60S, 40S, 60S composed of 28S, 5.8S and 5S rRNA, while this 40S is composed of 18S and near about 34 proteins. So this is the requirement of ribosome. It is present on endoplasmic reticulum. The endoplasmic reticulum is studded with the ribosomes, and these are known as factories for the protein synthesis. These ribosomes are known as factories for the protein synthesis. Then amino acid, near about 20 amino acids are required for the protein synthesis, and different factors are required. They are known as initiation factor, elongation factors and termination factor. So they are represented by the name, that is eukaryotic initiation factor, eukaryotic elongation factor and eukaryotic termination factors, or release factor is known as termination factor. Okay, so release factor, it is known as termination factor. Got it? It is responsible for the termination of protein biosynthesis. Then these are the various steps in the protein biosynthesis, that is activation of tRNA, or in other way it is known as charging of tRNA, then initiation of process, elongation and termination. So we'll discuss this one by one. So first is activation of tRNA. So tRNA is activated by the attachment of amino acid to the tRNA, that is synthesis of amino acyl tRNA. It is a two-step process. In first step there is activation of amino acid, and in second step there is attachment of amino acid to the acceptor arm of the tRNA. So we will discuss the activation of tRNA. So in the activation of tRNA there are two steps. First is activation of amino acid, and second one is the attachment of amino acid to tRNA. Okay, so in first step these amino acids with the help of ATP in presence of amino acyl tRNA synthetase. It gives enzyme AMP amino acid complex. Okay, this is activated amino acid. Now this activated amino acid, it is attached to the acceptor arm, that is CCA. It get attached with the acceptor arm. Suppose this is tRNA, so tRNA, this is CCA end, and to the hydroxyl group of the CCA end there is attachment of amino acid. This is known as charging of tRNA. So here charging of tRNA occurs. It is a two-step process, and the DHU arm of the tRNA, it helps in the recognition of this enzyme amino acyl synthetase, tRNA synthetase, DHU arm. It helps in the recognition of this enzyme. This is considered as proofreading step in the translation process. Got it? So it is a two-step process. First there is activation of amino acid, then this activated amino acid attached to the tRNA to form a activated tRNA or charged tRNA. Now second is the initiation. It includes, the first step in the initiation is activation of mRNA Second one is the formation of ternary complex. Then formation of 43S pre-initiation complex. Then formation of 48S initiation complex. And fifth one is the formation of 80S initiation complex. So these are the steps in the initiation: activation of mRNA, formation of ternary complex, 43S pre-initiation, 48S initiation, and 80S initiation complex formation. So we'll see one by one. Coming to the activation of mRNA. This mRNA in the eukaryote, it is activated with the help of different factors. So this is cap, this is 5' cap, this is poly A tail, 3' poly A tail. So it is activated with the help of factor 4F, which is composed of 4G plus 4A plus 4E. These are the factors. This is cap binding factor 4F. It forms the complex at the cap. So 4F complex is there, cap binding complex. So this E, 4E help in the recognition of the cap. Recognition of the cap. While this 4A, it removes the secondary structure in the mRNA. It removes the faults in the mRNA. Okay, so 4E help in the recognition. 4A removes the secondary structures, and 4G, it is a part of 4F complex which attached with the cap. Got it? Then after the formation of cap or attachment of 4F to the cap region, there is attachment of 4B and poly A binding protein. Attachment of 4B and poly A binding protein. So this results in the attachment of, this is 4F. This 4F help in the attachment of this mRNA to the ribosomal unit. Okay, this 4B, it helps in the recognition of AUG. It helps in the recognition of AUG, while this poly A binding protein, it helps in the recruitment of 40S ribosomal unit to the mRNA. Recruitment of 40S ribosomal unit to the mRNA. So this is active mRNA. This is active mRNA. This is 4F, that is cap complex. This is 4B, this is poly A binding protein. The sequence upstream of this AUG, upstream of the AUG sequence, which is complementary to the 18S rRNA, 18S rRNA or 18S ribosomal unit, which is complementary to the 18S ribosomal unit, which act as a consensus sequence for the recognition of this AUG codon. It is known as Kozak sequence in the eukaryote. It is known as Kozak sequence in the Kozak consensus sequence in the eukaryote. While same sequence in the prokaryote is known as Shine-Dalgarno sequence. In eukaryote, it is known as Shine-Dalgarno sequence, while in prokaryote, it is known as Kozak sequence. So this is activated mRNA. Once again, I will repeat the steps. There is 4F, formation of 4F complex, which is a combination of 4G, 4A and 4E. E help in the recognition of the cap. 4A remove if there is any secondary structure in the mRNA. 4G is a part of 4F complex, and 4F attached to the 5' end. Okay, which is 7-methylguanosine triphosphate end. Now the 4B, which recognizes the AUG bind with the mRNA and poly A binding protein. These proteins attached to the poly A sequence, which is at the end of the mRNA. It helps in the recognition of, and what is the role of poly A binding protein? It recruits the 40S ribosomal unit to the mRNA. It helps in the recruitment. Okay, and there is one sequence which is complementary to the which is complementary to the 18S rRNA sequence. It is known as Kozak sequence, which is in the upstream of this AUG and help in the recognition of this AUG codon. So this is all about the activation of mRNA. Then second is the formation of ternary complex. It is formed by the tRNA plus eukaryotic initiation factor 2 and GTP. Suppose this is GTP, this is eukaryotic initiation factor 2, and this is tRNA, that is methionine tRNA. This complex is known as ternary complex. Now moving to the third step, that is 43S pre-initiation complex. Okay, so we'll discuss that. So this 80S ribosomal unit, it is dissociate into 60S and 40S. This is with the help of initiation factor 6. It is with the help of eukaryotic initiation factor 6, and this unit the factor initiation factor 3 and initiation factor 1A prevent the reassociation of this small unit to the 60S ribosomal unit. What is the role of 6 factor? It causes the dissociation into 40 and 60S, while this reassociation, it is prevented by the factor 3 and factor 1 and factor 1A. Okay, then there is attachment of this ternary complex So this is factor 3, this is factor 1A, this is factor eukaryotic initiation factor 2 with GTP. Suppose this is GTP, and this is methionine tRNA. This is CCA. This is methionine tRNA. These are anticodons. Okay, this complex is known as 43S pre-initiation complex. Now this 43S pre-initiation complex is converted into so this is unit, this is 40S ribosomal unit, this is factor 3, this is factor 1A, this is eukaryotic initiation factor 2 with GTP. Okay, this is mRNA, this is 4F complex. Okay, this is poly A tail, this is poly A binding protein, and here is the 4B, factor 4B. Here is AUG. Okay, this is tRNA, this is methionine tRNA, 1 2 3. Here is the methionine. These are the anticodons. This is codon, this is corresponding region on the tRNA is known as anticodon. Okay, so this complex is known as 48S initiation complex. Okay, so ribosome, ATS ribosome, it dissociates into 40S and 60S with the help of eukaryotic initiation factor 6. Then 3 and 1A prevents the rebinding of this 40S to 60S unit. Then ternary complex attached with this 40S ribosomal unit form the 43S pre-initiation complex, and then mRNA attached to this complex, which is known as 48S initiation complex. Okay, now with the help of factor, that is eukaryotic initiation factor 5, the ATS unit will attach to this unit, and there is formation of, this is 60S unit attached to this 40S to form a ATS initiation complex. So it is composed of mRNA, then tRNA, this is methionine, these are anticodons, these are codons, AUG. So it is known as initiation complex, ATS initiation complex. Now this complex has 3 sites. So one is the peptidyl site, another one is the amino acyl site, this is A site, this is P site, and the third site it is known as exit site. A site for the attachment of the new incoming amino acyl tRNA, it is for the new incoming amino acyl tRNA. This is the site for peptidyl tRNA. Okay, this is the site, P site, it is for the peptidyl tRNA, and exit site, it is for release of tRNA, deacylated, release of deacylated tRNA. Okay, so these are the 3 sites, A site, P site and E site on the ATS initiation complex. So this is all about the initiation. So initiation, it includes the activation of mRNA with the help of different factors, then formation of ternary complex, then formation of 43S pre-initiation complex, 48S initiation complex and ATS initiation complex. Now elongation. Second one is the elongation. In elongation, there are 4 steps. The first one is the attachment of this amino acyl tRNA to A site. Second one is the peptide bond formation, peptide bond formation or elongation of peptide chain with the help of peptidyl transferase activity. Third one is the translocation of ribosome, and fourth one is the release of deacylated tRNA. So these are the 4 steps in the elongation process. Attachment of new incoming tRNA, amino acyl tRNA, it is with the help of energy. Okay, it is with the help of energy and with the help of elongation factor 1. With the help of elongation factor, that is known as eukaryotic elongation factor 1. Then peptide bond formation, which doesn't require any energy. There is bond formation between this peptidyl tRNA amino acid and amino acyl tRNA amino acid. Then translocation of ribosome occurs to the next codon and release of tRNA. The deacylated tRNA will remove from this exit site. So these are the 4 basic steps in the elongation. So we'll see the elongation. This is 4F. Now new incoming tRNA, so this is tRNA. Suppose this is incoming tRNA with second amino acid. This is initiator methionine. This is first one is the initiator methionine with second amino acid will attach to this side. Okay. This second will attach to this side. This is known as attachment of tRNA to A side. It requires eukaryotic elongation factor 1 and it requires energy. It requires energy, okay, for the attachment to the A site. Then there is peptide bond formation between these two amino acid occurs. Okay, so peptide bond formation between these two amino acid occur. So this is mRNA. So this is second amino acid and this is methionine. So peptide bond formation occur with the help of peptidyl transferase activity. Peptidyl transferase activity, which is present in the bigger unit of ribosome. Peptidyl transferase activity, it is present in bigger unit, that is 28S rRNA. It is present in 28S rRNA, peptidyl transferase activity. With the help of this activity, there is a bond formation between these two amino acid occur. Okay, this amino acid, that is methionine, it correspond with the N-terminal amino acid in the peptide chain. Okay, so there is peptide bond formation occur. Okay, now this deacylated tRNA, it enters the exit site. It enters the exit site and then released. Okay. So peptide bond formation occur. After that, translocation occur. This one is the AUG. This one is the another codon. Then next to codon. Then translocation of ribosome occur. It also requires energy. Okay. And the one factor is required, that is eukaryotic elongation factor 2 for the translocation of ribosome. So translocation occurs. In this translocation, this ribosome moves to the another position. Okay. So in this way, this amino acid, this tRNA, it enters this site. It enters peptidyl site. This is 2. This is methionine. And this A site is now free. This A site is now free for the attachment of new incoming amino acid tRNA. Now this site, A site, is free for new incoming tRNA. Okay, this is known as translocation. This process is known as translocation of ribosome. Okay, and this process, the attachment of tRNA to A site, peptide bond formation, then translocation and release of tRNA. This will repeat again and again until this assembly will encounter the stop codons. If there is stop codon, this will stop. Okay, this will signals the termination of translation process. So in termination, the release factor is important. Release factor 1 and release factor 3 and ATP. energy is required. Okay, and it requires stop codons for the termination. So you know the stop codons, that is UAA, UGA and UAG. Okay, so once it encounter the stop codons, it will recruit the release factor, which is structurally similar to that of the tRNA. Suppose this is release factor. To this release factor, there is release factor 1, there is attachment of release factor 3, and energy is required. Okay, so this will causes the hydrolysis of this peptide. Suppose this one is the growing, suppose this is methionine, this is second amino acid, this is third amino acid, this is fourth, this is fifth amino acid, and this is sixth amino acid. And when it encounters the UAA, there is termination of signal. So this termination, the bond between the tRNA and the amino acid, it is cleaved with the help of this release factor with peptidyl transferase activity. It causes hydrolysis of bond between the tRNA and the amino acid. So peptide release will occur when it encounters the release factor. So after the release factor, there is dissociation of this ribosomal unit into 60S and 40S ribosomal unit, then release of peptide chain, release of mRNA will occur. So all factors will dissociate after the release factor attachment. So energetics required for the peptide bond formation, there is no net energy is required, but it is for the formation of peptide bond, 4 ATPs are required. First 2 ATPs are required for activation of tRNA or charging of tRNA, and then next is required for the attachment to the A site, and next is required for the translocation of ribosome. So these are the 4 ATPs required for the peptide formation. Okay, 4 ATPs are required for the peptide formation, and this is the termination process. So it is divided into 4 main steps, that is activation of tRNA or charging of tRNA, then initiation process, then elongation and termination process. So initiation, it is discussed under the activation of mRNA, formation of ternary complex, formation of 43S pre-initiation complex, formation of 48S initiation complex and ATS unit. Then elongation in the form of attachment, peptide bond formation, translocation and release of tRNA, and termination with the help of stop codon, release factor 1 and 3, and energy is required in the form of GTP. Okay, so this energy which is required for the termination, it is not considered for the peptide bond formation, because it is not required for each and every peptide bond formation. It is required for the release of peptide after the synthesis of peptide chain. Okay, after a synthesis of polypeptide. So this is not under the calculation. So 4 required, and it is under the regulation of 2 main factor, that is eukaryotic initiation factor 2 and initiation factor 4E, which is in the 4F complex. These are the 2 checkpoints in the translation process. And proofreading with the help of amino acyl tRNA synthetase activity. The DHU arm of the tRNA help in the recognition of specific amino acyl tRNA synthetase. So this is required for the proofreading in the translation process. So this is all about the eukaryotic translation. So keep watching. Thank you.